Okay, so good evening, everyone. So today we are in the week 7 problem solving session of the NPTEL course, Genetic Engineering Theory and Application. So I hope all of you have gone through the week 7 lectures. I'll wait for 2 to 3 minutes and then I'll start today's session so that other learners can join. Okay, so in the last session, we were studying about the different expression vectors. So what are the characters of an expression vector or uh, what for an expression vector, what all sequences must be present in our vector, starting from the promoter. Then there should be a terminator region. Then between the promoter and terminator, we should have the gene sequence. And also, if the gene of interest. So we were looking at the what are the characteristics of an expression vector, how is it different from a cloning vector, and for basically expression vector means if you want to express the gene of interest, that is we want the gene to be transcribed into mRNA, and then this mRNA will be further translated to protein. Okay. For an expression vector, we will need a promoter. We will need a promoter so that the RNA polymerase can bind and transcribe and start transcription of the gene of interest. Then terminator is required so that at the terminator region, the RNA polymerase will uh, will uh, will get removed from the genomic sequence, isn't it? And then we will have the mRNA. So once we have the mRNA, then this mRNA also needs to be translated. So this expression vector should also have a ribosome binding site. Okay, which in case of this in prokaryotes, we have this Stein-Dalgarno sequences. and this ribosome binding site will be present in the 5' untranslated region of the mRNA. Okay, and at this region, the ribosomes will bind and translate the mRNA to protein. Okay, so and also we were looking at the what all other things can be present in the expression vector. So depending on which is the host system, whether it is bacterial or yeast or mammalian, we have to use those type of expression vectors. So there can be mammalian expression vector uh, if we want that the gene to be expressed in a mammalian host. If we want it to be expressed in a yeast host, then there are yeast expression vectors. If it is a bacterial host, we usually go for the pet vector system. Okay. And also, if we want to purify that protein further, then we can also add the fusion tag to the gene of interest so that we can purify it. And also the fusion tag, sometimes they can be used to make our protein soluble. Okay, so I'll start today's session. So okay, so yeah, hello everyone. I'm Akash Jyoti Pathak, and I'm one of the PMRFTs for this NPTEL course Genetic Engineering. And this course is being run by Professor Vishal Trivedi of IIT Guwahati. So let's first have a quick recap of what all we learned in week 7. Yeah, in week 7, we are mainly learning about the downstream processes, isn't it? So downstream processes means what? So once we have expressed the protein of our interest, okay, once we have expressed the protein of our interest, then the next step would be what? Isolation of the protein. So first we need to isolate the protein from the cell, from the host cell. Okay, so there will be there are different methods by which we can. So how we can isolate the protein from the host cell? Basically, we have to lyse the cell, isn't it? We have to lyse the cell. So so there are two cases. If it is an extracellular protein, then there is no requirement of lysing the cell, isn't it? Extracellular protein means our protein is getting expressed in the media. Okay, and if it is an intracellular protein, intracellular protein means the protein is expressed and it is present inside the bacterial cell, okay, or inside the host cell. So in that case, we need to lyse the cell first. So there are different methods of cell lysis. Okay, so what are the different methods of cell lysis that we learned in week 7? So there can be physical methods. Okay, there can be physical methods, chemical methods, then there are enzymatic methods. So these are things you we all have learned in class 12th as well, isn't it? Basics of this. So enzymatic methods, and then there are also mechanical methods. Mechanical methods can be considered a part of physical methods as well. Okay. So once we have lysed the cell and we have obtained the protein, the next step would be to purify the protein. Okay. Because the bacterial, when you are, for example, you are expressing your protein in a bacterial host cell. Okay. In a bacterial host cell. So your bacteria will not only express your protein of interest, but it will also express its own protein. Okay, all the different proteins that is required by the bacteria for its own survival, for its own metabolism, for its own growth. So such type of there are constitutive proteins, that are proteins which are expressed all the times. Okay, so such type of proteins will also be expressed. Now, from all these mixture of proteins or all these different types of proteins, we have to purify our protein of interest. Okay, so there are so how we can purify the protein of our interest by the technique called chromatography. Okay, so we also learned about the different techniques, chromatographic techniques. Okay, so different. What are the different chromatographic techniques? Anyone? So the different chromatographic techniques are there are ion exchange chromatography. Okay, so the basic principle of chromatography is what? Ion exchange chromatography. Then there is size exclusion chromatography. Then there is hydrophobic interaction chromatography, and then there is affinity chromatography. Okay, there are four different methods of chromatography, and all these techniques, all these chromatographic techniques are used to purify our protein of interest. Okay, and in all these techniques, what are we doing basically? We are uh, the basis is the character of a protein. What is the size of a protein? What is the charge that is present on our protein? What is the hydrophobicity of our protein? What is the affinity? Affinity means if we have added any fusion tags like 6x histidine tag, or we have fused a say a GST tag. We have fused the GST tag to our protein of interest. So based on the affinity of that protein to the column matrix, so we are utilizing the different properties of a protein, its charge, its size, or its hydrophobicity. Okay. So let's see one by one. So yes, upstream processes. In upstream processes, so in all the earlier lectures, we learned about the upstream processes, starting from the cDNA library, genomic library, how we can identify the gene of interest. So now when we have identified the gene of interest, we restriction digest the gene of interest, and we also restriction digest the do a restriction digestion of the vector, and then we clone our gene of interest inside the vector. Now, once we have the recombinant plasmid or the ligated plasmid, the next step is we transform this recombinant plasmid inside the host cell. So once we have transformed it, the next step is we screen for the cell which has taken the recombinant plasmid. Okay, so we learned about the different methods by which we can screen for the recombinant recombinant cell. Okay, now, so this is the screening. So these are the upstream processes. Now, once these all things have been done, in the next step, what we will do, we will express the protein of interest. So after the protein of interest is expressed, now this protein can be an intracellular product or it can be an extracellular product. So if it is an intracellular product, then we have to go for different cell disruption methods. So that is what I was explaining. So what are the different cell disruption methods? Chemical, enzymatic, mechanical, and physical. And if it is an extracellular product, then we can directly go for the purification of our product. Okay, so there are different methods. There's solid liquid extraction. So this extraction, filtration, centrifugation, these methods we can use. And once we have extracted the protein of interest, then we concentrate the protein. Okay, because we want a small or a large amount of protein will be purified, right? So from that we can concentrate it to get a high yield, and then we can use it for further processes. Okay, so purification, and we get the final product. So what are the different disruption methods? So the host cell, basically the host cell has different characteristics, isn't it? So it has a physical. Okay, so the host cell has different characteristic. It has a physical strength, it has a chemical composition, and it has a structural organization. So, utilizing this different physical strength or these different characteristics of a host cell, we use different cell disruption methods. So, the host cell can be a prokaryote yeast, it can be an animal, it can be a plant cell. And if you look at all these different types of host cells, these different host cells have different physical strength, isn't it? Your animal cell doesn't have a cell wall. Your plant cell has a cell wall. And if we look at the cell wall again, then the cell wall of a plant cell is different from the cell wall which is present in yeast and cell wall which is present in prokaryote. Okay. Similarly, if you look at the structural organization in these different host cells, they are also different. So plant cell has a large vacuole, your animal cell has a small vacuole, and then there are other components which are present in plant cell and that are lacking in animal cell, isn't it? Their chemical composition, structural organization, all these are different, and utilizing these differences, we have to use different cell disruption methods. So what are the different physical, what are these different methods? It can be physical methods. In physical methods, you have thermolysis, osmotic shock, sonication. Chemical methods, we have alkali treatment, detergent treatment, or permeabilization. And there is enzymatic methods. Okay, enzymatic methods means utilizing the different enzymes which can be used to digest the cell wall so if it is a plant cell, we can use pectinase or uh, there are the different components which are present in the cellulase. So cellulase and pectinase, all these enzymes can be used. If it is an yeast cell, we can use chitinase. If it is a bacterial cell, we can use lysozyme. Okay, so these are the different enzymatic methods. And in mechanical methods, we learned about the bead mill and the high pressure homogenizer. Okay, so these things are easy to understand, isn't it? So so we'll quickly go through this. Physical methods is thermolysis, means what you are giving a heat shock. Uh, so so this method is easy, economical, and require no additional specialized equipment. But thermolysis has a disadvantage that this method can only be used if the product is thermostable. Okay. So if you want to isolate the protein from your host cell, and your protein, and the protein is very unstable. So you are keeping it at temperature of 95 degrees Celsius to lyse the bacterial cell, and the protein that you isolated, that got denatured at such a high temperature. So that will be of no use, isn't it? So this method gives a heat shock to kill the organism, and as a result, it disturbs the cellular integrity without affecting the product. So the effect of heat shock depends on ionic strength, presence of chelating agents such as EDT, and presence of other proteolytic enzyme. So along with heat shock, you also need to provide different types of buffers. So it must have EDTA and all these, so that the RNAs, DNAs, all these enzymes that are that once you lyse the cell, you will have all these different enzymes as well, proteinase. So you do not want these DNAs and RNAs to interfere with your protein of interest, so you can add EDTA that will chelate out these DNAs and RNAs, and also you can add different types of proteolytic enzymes so that other proteins gets degraded while you have your protein of interest. So now osmotic shock, we all know, isn't it? So what is osmotic shock? So if we place a cell in a hypotonic medium or a hypertonic medium, then what will happen? So if we place our cell in a hypotonic medium, then what will happen? Inside the cell, you have a lot of salt concentration, and outside you have less salt concentration. So what happens is the water solvent molecules which is present in the environment will move inside the cell. And as a result, what will happen? Your cell will swell. Cell will swell, and after keeping it uh, at a particular salt concentration, which is which has a salt concentration lesser than the salt concentration which is present inside the cell, your cell will rupture. Okay, your cell will rupture, and all the contents which are present inside the cell, it will come out. Okay, this method can only is usually used for mammalian cell, isn't it? Because mammalian cells do not have a cell wall, so you can use this method for uh, for extracting the contents within the mammalian cell. So maintaining an osmotic balance is an active process with expenditure of energy. So prolonged exposure to the cell with hypotonic liquids such as water. If you place a cell in water, then what will happen? If you place your cell in water, then your water is having very less salt concentration. So all the water which is present in the environment will move inside the cell. And what will happen? Your cell will swell and ultimately burst to release the products. So according to the Van Hoff's equation, osmotic pressure pi is directly proportional to the concentration of the solute and temperature. So we all know this formula, isn't it? So what is the osmotic pressure? That will depend on what is the gradient, concentration gradient, or what is the difference in concentration inside the cell and outside the cell. So that is Ci minus Co. So difference between the solute concentration inside and the outside the cell. Okay, so when there is a difference in this concentration gradient inside the cell and outside the cell, the water molecules will move inside the cell, and after a certain time, what will happen? An equilibrium will be reached when there will be the molecules are either going out or they are either coming out. So net movement is zero, isn't it? And it also depends on this absolute temperature and universal gas constant R, isn't it? That is the osmotic pressure, and each mammalian cell has different susceptibility towards osmotic shock. So red blood cells, they are very prone to lysis, and even a tiny drop of water will lyse the cell, while plant and bacteria cells, they are more resistant towards osmotic lysis because they have a thick cell wall. Okay. So these points are clear. Now another method is sonication. So what is done in sonication? So this is basically a probe sonicator. Okay. So if you have worked in any lab, you must have seen this. So probe sonicator this is the probe. Okay. This is the probe of the sonicator, and this probe of the sonicator, this produces ultrasound. Okay. Ultrasound. So, and what is this? What is the frequency of the ultrasound? It is 20 kilohertz. And the what is done is this bacterial cell or whatever cell you have, mainly this bacterial cell, you resuspend the bacterial cell pellet, and then you insert the probe inside the resuspended bacterial cell pellet, and then you switch on the instrument. So this probe will produce this ultrasound, and what happens as a result of ultrasound? It produces compressions and rarefactions. So this alternating compression and relaxation. So it will produce compressions, and then there will be relaxation. And as a result, the water molecules, what happens if the air bubbles are produced in the media. Now this air bubbles gradually grow in size. Okay. These air bubbles grow in size, and after a certain time, these air bubbles explode or implode. So when these air bubbles explode, the the energy that is released, that causes the lysis of the cell. Okay, so this is basically the figure. You can see this figure. So this is the ultra sound that is produced by the probe sonicator. So there is compression and relaxation, and as a result, there is formation of bubble. Now these bubbles gradually grow in size. Now after a certain time, this bubble will attain an unstable size. And then there will be bubble implosion. The bubble will burst, and there will be a large amount of energy is released. Okay. And this energy is utilized for slicing the cell. Okay, so that is called cavitation basically. Sonicator generates ultrasound waves of frequency more than 20 kilohertz to cause cell disruption by cavitation. Cavitation means basically this ultrasound is producing air bubbles, and the interaction of ultrasound with liquid causes compression and decompression very rapidly. The bubble formed in liquid. So the bubble formed in liquid, this process is called cavitation. It compresses several thousand atmospheres and gives shock waves to the cell wall or plasma membrane to cause cell lysis. Generation of ultrasonic waves in liquid causes rapid change in temperature and may cause thermal denaturation. So what is done is whenever we are lysing the cell by sonication, we place this cell lysate we place this bacterial cell in a ice, inside ice. Okay, because of this process, a lot of heat is generated. So you, as a result, you must once your protein is isolated from the bacterial cell, you must provide a low temperature. Otherwise, your protein will get denatured. So we always keep this resuspended cell pellet, bacterial cell pellet inside the ice, inside an we take an ice bucket and we fill it with ice, and when we place this cube inside this ice bucket, otherwise the temperature will increase by several times. Okay, then there are different chemical methods. So these things are they are not very difficult to understand, isn't it? So physical methods, and what are the different physical methods that we learn? So one is this thermolysis, second is osmotic shock, and third is sonication. Okay, now in chemical methods, alkali treatment is what? So this is harsh but effective chemical treatment to lyse the cell. So all these different methods depend on the type of cell that you want to lyse. Okay. So alkali treatment causes lipid saponification, which disturbs the lipid packing and affects the cell wall integrity. Then there is detergent. So detergent is when we are using these type of chemicals like SDS, CTAP, Triton X100, saponin, digitonin. So all these will work on the principle of critical missile concentration. So what it does is, as you have seen, if you have seen the structure of a detergent, it has a hydrophilic head and a hydrophobic long tail. So if you look at, so say for example, sodium dodecyl sulfate, it has a very long hydrophobic chain. So dodecyl means it has a 12 hydrocarbon, 12 carbon hydrocarbon chain. And then it has a sodium ion at its head group. So because of this hydrophobic tail and hydrophilic head, it will interact with these membrane proteins. The membrane proteins which are having this they will also have an extracellular domain and intracellular domain. So this extracellular domain will be hydrophilic and this intracellular will be hydrophobic. So these detergents basically it will form a micelle around this protein molecules. It will okay. Uh, The detergents will form a micelle around these protein molecules and it will solubilize it. Okay, cause solubilization of the, it will also cause solubilization of the lipid because lipid also has this hydrophobic head and hydrophilic tail. And the effect on increases linearly with concentration. The detergent concentration, which causes abrupt change in lipid solubility and forms micelle, is known as critical micelle concentration. So as you are increasing the concentration of the detergent at a particular detergent concentration, the detergent will form a micelle around this lipid molecules and the solubility will decrease. Okay, and it will get precipitated. So examples of this detergent SDS, CTAB, and Triton-X. Another method is permeabilization. So a partial cell wall disruption or permeabilization is achieved by organic solvents such as toluene. The organic solvent is absorbed by the cell wall, resulting in its swelling and ultimate rupture. But at low concentration, organic solvent permeabilizes the cell wall without disturbing the cellular integrity. So this process allows to use cell as a reaction vessel to catalyze a reaction and get desired product. Okay. So these organic solvent will be absorbed by the cell wall and it will swell. And if at a particular if a particular concentration of this organic solvent is used, then the cell wall will only get ruptured and your cell will remain intact or the contents of the cell will remain intact. So enzymatic methods, these are the different enzymes like lysozyme is used for bacterial cell lysis. Okay. Then other than lysozyme, there can be glycosidases, acetylmuramyl, L-alanine, amidase. So if we were discussing about the structure of peptidoglycan, it is made up of alternating NAGNAM units. So for digestion of these subunits, we can use these enzymes, endopeptidases. Then for each cell, we can use glucanase, protease, manganase, chitinase. So depending on the composition of the cell wall. So we can use, there are different enzymes. Yeast cell wall, yeast is basically a fungi. So we can use chitinase. The fungi cell wall is made up of chitin, so we can use chitinase. Then there are different type of carbohydrate groups which are attached on the cell wall, like mannans or these sugar groups are present. So we can use glucanase, mannanase, all these. And for in most of the enzyme-mediated cell lysis method, the rupture of cell wall depends on the osmotic pressure of the external medium. In few cases, enzymatic digestion is performed to remove external, and then in the sensor is the okay. So basically, what is done in enzymatic methods is once you have harvested the cells, you have harvested the cells means you have grown the culture, and then you have centrifuged it, the culture at 6,000 RTM for 10 minutes, and you have obtained the bacterial cell pellet. Now, once you have obtained the bacterial cell pellet, yeah, you resuspend this bacterial cell pellet in a lysis buffer. Now, what you do is in this lysis buffer, you add the enzyme. Okay. So, for bacterial cell wall lysis, we usually add lysozyme and then followed by treatment with lysozyme. After that, we go for sonication. Okay. So basically, your enzymatic digestion is only digesting the cell wall. It is only digesting the cell wall, but you also need to rupture the cell membrane because your the protein or all the contents is present inside the cell membrane. So for rupture of the cell membrane, we have to again go for any other physical methods like sonication or osmotic shock or any other methods or heat shock method we can also do. Okay, so thermolysis basically high temperature is given and then followed by that low temp. Again, it is thawed at very low temperature. You flash freeze it in liquid nitrogen. Then again you keep it in water bath at 42 degrees Celsius. Then again it is flash frozen in liquid nitrogen. So this is done in several cycles again and again. So this freeze thaw, repeated freeze thaw cycle will rupture your cell wall and or the cell membrane and uh, basically it ruptures the cell wall and then you have to again go for sonication to extract the contents which is present in the cell. Okay. Then there are these mechanical methods. So in mechanical methods, one example is this bead mill disruption. So in bead mill disruption, you have this you have different beads which is present in this chamber, and there is an impeller. So this impeller will there is a rotating shaft and this rotating shaft will rotate the impeller and this is the cell suspension. You pour this cell suspension bacterial lysate, you have resuspended it in a buffer and then you have poured it inside this cell pellet and then this impeller rotates and these different beads they will interact or with the cell and it will grind the cell. Basically, it is similar to your motor pistol that you use. Okay, motor pistol or these different mechanical methods of cell lysis. We are applying force and pressure to rupture the cell wall. So that is the mechanical method. So it is so this is yeah it has a grinding cylinder. It has a central shaft with number of impellers. And this impeller can rotate clockwise, anti-clockwise. And the grinding cylinder is filled with beads made up of glass, alumina, titanium carbide, zirconium oxide. There is an inlet to supply the cell suspension and an outlet to collect the sample after the process. So when the bead mill runs, the cell experiences shear force. So these will experience a shear force, and this shear force will be exerted by these glass beads, these different beads, and as a result, it will force open the cell. Okay, then so these are some of the information that you must have. So the rate and degree of cell disruption will depend on what? It will depend on what. So all these different methods are depending on what type of cell you want to lyse. So that will depend on the cell type, what is the thickness of cell wall, what is the localization of the product. So this protein can be either be present in the cytosol or it can be present inside inclusion bodies. So depending on that, you have to go for several other rounds of disruption. So if your protein is present in the inclusion bodies, you need to add detergents as well. This Triton X treatment is required, and then you have to go for solubilizing this inclusion bodies by guanidinium hydrochloride, and then you will get your protein. Okay, so that is there are different factors. It will depend on the localization of the product, agitation, speed of impeller, bead size, density and loading, residence time and temperature. So the cell disruption in a bead mill and release of product is a first order kinetics, and it may be given by the first order equation. Okay. So you know the first order equation ln A minus ln A naught equal to KT. Okay. I can write this ln A by A naught. A naught is the initial concentration. So if you express it, okay, then you will get this equation ln C max by C max minus C. So Cmax is the concentration of product that can be released from a given amount of cell suspension, and C is the concentration of the product released at a given time t. So K is the first order constant. So the Cmax is the maximum concentration of product that can be released, and the what is the concentration of product that is released at time t, that is given by C. So from this equation we can get that what concentration of product can be obtained. And this value of K will depend on the different properties of this bead mill. What is the type of impeller, what is the bead size and loading. So you remember that it follows the first order kinetics, the cell disruption in bead mill and release of product. Then there is this high pressure homogenizer. High pressure homogenizer is also commonly called French press. Okay. So these French press is also present. They're in different laboratories. So there is a basically you have this instrument, and then there is a chamber inside this. So you pour your cell lysate in this chamber, and then there is a steel metal rod like cylindrical object, and you press this. You press this. This. So this is what actually. So this is this cylindrical object is is forced into this space that is there, and what happens, the cell lysate that is there, that cell lysate is uh, the cell lysate will pass through this small valve, and it will the high pressure that is produced, that will force open the cell. Okay. So this high pressure homogenizer consists of a high pressure positive displacement pump connected to an adjustable discharge valve, and the cell suspension is sent into the homogenizer through a small homogenizing valve at a very high pressure. So this cell is made to this lysate is made to pass through this valve at a very high pressure, and because of the very small pore which is present, your cell will be it will experience a very high shear stress, and this shear stress will cause the cell to get lysed. Okay. So you can use different methods in depending on what you have in the lab. So if you want to lyse a bacterial cell, few labs have sonicator, few labs have French press. So if you have a sonicator, then you can go for using a sonicator. If you have a high pressure homogenizer, you can use this French press. And also it depends on the what is the lysate volume. So if it is if the lysate volume is very high, then using a French press is advantageous over using a sonicator. So in sonicator, because this size of depending on the size of the probe, you cannot have a very large volume of cell lysate. Okay. So what is this? It experiences a shear stress, isn't it? So what is the shear stress that the cell experiences, that is given by this equation? PS equal to half rho V square. This is rho. That is the density of the fluid, and PS is the dynamic pressure, and V is the jet velocity. So the small valve that is present, the lysate comes out from this small valve with this velocity, and the shear force that it will experience will depend on the velocity with which it is ejected, and the density of the fluid. So cell disruption in high pressure homogenizer, that is also a first order kinetics reaction. Okay. ln C max minus C max by C max minus C equal to Kn. So what is N here is the number of passes through the valve. So what is done is basically once you so if you see this figure, you pour this feed inside this chamber, and then you press this cylindrical object, and a high pressure is created, and this cell comes out from this small orifice which is present in the valve. Now the lysate that you got, that is again loaded into the feed. So this is done around 2 to 3 times. So the lysate, the homogenized product that is obtained, that is again poured into the feed. So it requires 2 to 3 rounds for proper lysis of the cell. Okay. So that is what. So n is the number of passes. That is why this factor comes n. The through the valve, and k is the first order constant. As high pressure homogenizes passes the cell at a very high speed through a narrow valve. It disrupts the cell, and simultaneously it lowers down the temperature as well. Okay, so everything is coming out from a very small port. Then what happens? The temperature decreases. So sonication. This is one of the advantage of using a mechanical press or high pressure homogenizer or a French press over a sonicator. That this sonicator, one of the disadvantages is that it produces a very high temperature, and uh, you need to keep this lysate um, inside the inside the ice, or you have to keep an ice always you must have an ice bucket, and you have to put your tube inside this ice bucket. While here in this case the problem of temperature is reduced considerably. Okay, so now answer these questions. The cause of cell lysis. What is the cause of cell lysis? During osmotic shock is what? Osmotic shock, what is happening? There is inflow of water, isn't it? So you place your cell inside in a hypotonic medium. Okay, in a hypotonic medium, and inside the cell there is a very high concentration of salt compared to the salt concentration which is present in the surrounding media. So your water will move inside the cell and it will lyse the cell. Okay. The enzyme commonly used to lyse bacterial cell. What is the enzyme commonly used to lyse bacterial cells? That is lysozyme. Okay. Lysozyme. Now the method involving a first order kinetic relationship for cell disruption is what? So all this mechanical disruption method that we learned, right? So the mechanical disruption method. So what are the two mechanical disruption methods that we learned? One is this bead mill method, and second is the high pressure homogenizer or French press. So all these follow the first order kinetics. Okay, they will follow the first order kinetics. So the answer is bead mill disruption. So osmotic shock is what? Osmotic shock is physical method. Thermolysis is also a physical method of cell disruption. Alkaline treatment is a chemical method of cell disruption. So the mechanical method of cell disruption will is usually follows the first order kinetics, reaction kinetics. Now the critical missile concentration, what is it? So it is that that particular concentration of detergent which will cause abrupt change in lipid solubility, isn't it? So you see here, so this is your cell membrane. So your cell membrane has all these phospholipid bilayer. So this is the phospholipid bilayer, and cell membrane also has the integral membrane protein. So so what is done? These detergents are added. So once the detergents are added, these detergents will form a missile structure around around the integral proteins, and also around the phospholipid bilayer. So your the solubility of this lipid will change abruptly, and the contents within the cell can be extracted. Now the enzyme that is used to lyse yeast cell is what? Which enzyme will be used to lyse yeast cell? That is chitinase, isn't it? Because the yeast cell wall is made up of chitin. It is made up of chitin, so you can use the enzyme chitinase for lysing the yeast cell. Or there is imolase also we learned in the last session. Okay, now let's learn about, so once you have this, so you have your cell, you have disrupted your cell. Okay, disrupted or you have lysed your cell. Now, once you have lysed your cell, then what will happen? All the contents within all the protein molecules which is present inside the cell, they will come out. So the next step will be what? To not only the protein, all the DNA, the RNA molecule, these lipid molecules, then there is this carbohydrate, all the biomolecules which are present inside the cell, they will uh, they will all come in your cell lysate. So that is called your cell lysate. Now from all the cell lysate, you have to purify your protein. Okay. So how will you do that? So purification, or it will basically follow what is the principle of separation that we need to understand. So for example, in this figure, which is taken from this lecture slide, you can see that these are the three different chemical compounds. One is benzene, one is phenol, and another is aniline. Aniline. Okay, so all these chemical compounds have, what are the difference? What are the differences among these chemical compounds? So they differ in the functional groups which are present on them. So phenol has OH functional group, aniline has a amino or the amine group, which is present as a functional group. And also as a result of these different functional groups, they have different physical and chemical properties. Okay, so what are the different physical properties? So if you see their molar mass are different, the density is also different the density of these three different compounds. These are the different physical properties that are different. Melting point, the boiling point. So if you see the melting point or the boiling point of phenol is very high. It is 181.7 compared to benzene, which is 80.1. So using this different physical properties of the compounds, you can separate one compound from the rest of the compounds. Okay, so these are the different physical properties: molecular weight, boiling point, freezing point, crystallization, solubility, density. And the chemical properties, what are the different chemical properties is they are having different functional group. And because of the presence of the different functional group, they have different reactivity. The reactivity towards different agents is different. Okay. So for example, if you want to separate benzene from aniline, uh, then what will you do? You can use both the physical and chemical properties of benzene into account and isolate it from the mixture. So you can use both these properties. You can either utilize the difference in physical properties among these compounds, or you can either utilize the difference in chemical properties. So depending on, so for example, if there is a chemical reactant which only reacts with aniline but not reacts with phenol, then you can use that chemical reactant, so that once this chemical reactant reacts with aniline, it will form a complex and it will precipitate, and then you can isolate the phenol. Or you can use the different physical properties. Like here in this example, you look at this figure. So you have a mixture of benzene and aniline, but the difference, what is the physical property that is different in benzene and aniline? It is the melting point. The melting point of benzene is much lower compared to aniline. So what will happen when you heat this mixture? At 80 degrees Celsius, what will happen? When you're heating this mixture at 80 degrees Celsius, your benzene will evaporate. Okay, it will evaporate and it will generate fumes. Now these fumes will pass through the condenser. So in this condenser, what happens? We have learned all these techniques, right? In class 7th, 8th, we have seen this. So when this benzene vapor passes through this tube, the which is present in the condenser, and in this condenser you have water inlet and water outlet, so which keeps the temperature of condenser very low. Then once it will move through this tube, the temperature will be lowered. Now, once this temperature will be lowered, this benzene will be converted, vapor will be converted into liquid. and this liquid can be collected. Okay. But because the temperature of aniline is very high, 181 degrees Celsius, this aniline will not evaporate. It will stay as it is. And by heating it at 80 degrees Celsius, we can separate the benzene from aniline. So what is distribution coefficient? So distribution coefficient is used to describe the distribution of compound 1 between two phases, A and B. So what is the concentration of compound 1 in phase A by concentration of compound in phase B? So that is distribution coefficient. Okay. So for example, there will be a distribution coefficient of benzene, that how much benzene is present in this phase A and how much is present in phase B. Okay. Now, another technique that is done for biological molecules. So these are chemical compounds, isn't it? So for biological molecules, we'll go for chromatography techniques. So what is the meaning of chromatography? So the purpose of chromatography is to separate a complex mixture into individual components, exploiting the partition coefficient. So, so basically in chromatography, you have two phases. Okay, but you have a mobile phase and you have a stationary phase. Stationary phase. Now, what is done is basically your analyte, whatever analyte you have. Analyte means the molecule which you want to purify. Okay, so that is allowed to pass through the stationary phase. Okay, your analyte will be allowed to pass through the stationary phase, or it will be injected inside the system, and your system has a column. So your analyte will interact with the stationary phase. Now, after your analyte has bound to your stationary phase, your mobile phase is passed. Now, now this analyte will have a particular affinity for the stationary phase, and it will have a particular affinity for the mobile phase. So if the analyte has more affinity for the mobile phase, then your analyte molecule will get eluted from the stationary phase. While if it is having more affinity for the stationary phase, then your analyte will remain bound to your stationary phase. So for eluting the analyte from the stationary phase, you need to change the mobile phase. You need to increase the concentration of mobile phase, or you need to increase the pH or the salt concentration of the mobile phase, so that you can purify your protein. So basically you have a column, and in this column you have different resins, or so this is called the matrix of the column. So this matrix of the column has resin. Now this resin is it can be a gel that is made of say for example there are dextran columns which is having dextrose and agarose linked to each other to form a mesh-like structure. Okay, or it can be a cefacryl, cephalose. So there are different types of matrices that are commercially available. So for example, cefacryl has this acrylamide molecules are cross-linked with another type of um, polymer. Okay, so these polymer matrices have different pore size. Okay, or they can have a particular charge which is present on the resin. So this is your basically this is your stationary phase. Okay, now you will pass the mobile phase through this stationary phase. Okay. Now, depending on the partition coefficient or the distribution coefficient, that what is the ability of the analyte to be distributed in phase A or phase B. Depending on that, your protein or your molecule will get eluted. So the phase A is stationary phase or matrix, and phase B is the mobile phase or buffer. You have a buffer. So this buffer is your mobile phase basically. Okay. So now column chromatography. So the stationary phase is filled into a cylindrical tube. So that is what I was explaining. So you have a column. So this is your column is basically a cylindrical tube, and you can see this in this figure that this cylindrical tube is filled with different resin or matrix. It has a particular matrix. Means it has a it can have different mesh like it has a mesh like it can be polyacrylamide agarose combined matrix or which will have a particular pore size, or it can have different type of matrices basically. So or it can have a particular beads which are having a which is giving a particular size, pore size. Okay, now the mixture of analyte. So this is your analyte. Your analyte has different molecules. This analyte is loaded into your column, and then your mobile phase is passed. So the mixture of analyte is loaded into the top, and it runs from top to bottom. So I assume so here they have considered two molecules X and Y, and it has a this is the X has a KD value, the Y will also have a KD value. So for example, X has a KD value 1 and Y has a KD value 9. So as they will travel, X and Y will partition between stationary and mobile phase. So because Y is having a higher KD, KD means what? The concentration of the analyte in phase A divided by its concentration in phase B. So if your phase is the stationary phase, if phase is the stationary phase and your KD value is 9, it means that it is having more affinity for the stationary phase. Okay, so your your Y will remain associated with the matrix while your X will be diluted earlier. Okay, it will remain associated with the matrix and remain on the top of the column while your X will move along the water. So this is your Y molecule because it has, and this is your X molecule. Your Y has higher KD value. Okay, higher KD value means it has higher affinity for the stationary phase over the mobile phase. That is why your Y will be uh, eluted later as compared to your X molecule. Okay. So now I will get you. So what when a chromatography experiment is done, you basically get a get a chromatography profile. That is the profile chromatograph profile that you get. That is called chromatogram. Okay. So the chromatogram is basically a graph. So this is a graph in which you have the basically you have an absorbance 280 on the y-axis and you have the time or volume on the x-axis. You can either have time or you can either have volume. And you and in this graph you can see there are different peaks. You will observe in this graph. So what is a chromatogram? So chromatogram is basically a graph which has the absorbance 280 on the y-axis and the time or volume of buffer passed on the y-axis. Okay, or time at which you are getting the eluent that is the present on the x-axis. So and you are having different peaks. So now what will happen? So these are called the different retention time. So the time at which a particular molecule is eluted, that is called the retention time. So for A molecule, your A molecule is getting eluted at this time. So this time is called TR1. The B molecule is getting eluted at this time. This time is called the TR2. Okay, similarly, this molecule is getting at another time. So that is the retention time for this molecule. Similarly, for C and D, you will have a different retention time. So as a result, because these molecules are having different KD values, okay, these molecules will have different KD values, so they will get eluted at different times. So you will have different retention time for each of these molecules. Okay, is it clear? So now there can be some molecules which are very different in their properties. So they will be their peaks will be very separated. So if you see this A and B molecules, their peaks are very separated. But for C and D molecules, they are overlapping, isn't it? They are not separated uh, as or they have not been separated very nicely as the A and B molecules have been separated. Okay, so they are fused peaks. So that is a chromatogram. So the plot of elution volume along with the absorbance is known as chromatograph. So you can have different detectors. So actually this absorbance at what wavelength depends on the detector. So if it is a you can have a absorbance 260 as well. So if your detector can measure the absorbance at 260 nanometer, then it will be a 260 detector. It can be a 280 detector. So depending on what type of detector you have, so it will basically it is giving the absorbance. Okay, and your x-axis has the retention volume at the time. Now, another factor, yeah, so these are different mathematical terms, several mathematical terms. So resolution is what? So resolution is basically how well you are able to separate the two peaks in a chromatogram. So you are seeing these different peaks. So how well you have been able to separate one peak from the other, that is called your resolution. To separate two analyte peak from another is known as resolution, and it is defined as the ratio of the difference in retention time between the two peaks and the average of base of peak width. Okay, so you see here, okay, so see this figure. So if you want to calculate uh, the resolution of peak A and peak B of analyte 1 and analyte B. Then how will you calculate? So you know the retention time of B. So you can minus TR2 minus TR1. And then you can, so you have this peak width. So this has a particular peak width. This has a particular peak width. You can take the average of the peak widths. So the average, so this distance say D1 and this distance say D2. So you can take the average of this, so that will be your resolution. Okay, so resolution is the ratio of the difference in retention time between the two analytes divided by the average of the peak width of the two analytes. Okay, average of the base of peak width. So remember this formula, and then so when resolution is 1, then what does it mean? So the separation is very good, isn't it? The separation of two peaks is 97.7%, and a column with RS more than 1.5 is considered good. So the higher the resolution value means that column is better, because that column is able to separate very similar molecules very efficiently. Okay, so the number of distribution events govern the ability of a column to separate the two analytes. In other words, resolution is directly proportional to the number of distribution events. Okay, so there is one concept of theoretical plates that has not been covered in much detail, but just understand this, that in column chromatography there is a so it is considered this column has several planes. And these planes participate in distribution of molecules. So this plates that are there, these plates have a particular height, and there are a number of there is a the number of theoretical plates and the height of each of these plates will be uh, so the height is h and the length of column is L. So the number of distribution plane in a column is given by n equal to L by h. So basically you have your column. Okay. So now if you imagine that your column has different theoretical plates. Okay. So each of these theoretical plates are having a particular height h. Okay, and this is the length of your entire column. Okay, L is the length of your entire column. So how will you calculate that how many theoretical plates are present in the column? So this is actually a concept. Okay, so if you imagine that there are several theoretical plates which are packed inside this column, then number of theoretical plates will be what? It will be L by H, isn't it? The length of the column divided by the height of each theoretical plate, that will be n. Now, this n, the number of theoretical plates basically determine what is the resolution of the column. So these are different parameters which are used to define the resolution of a column. What is the this? So you have different columns. Say for example, so what is the resolution of one column? How can you express it? So these are different parameters. Okay. So these are things that are to be considered when you're trying to construct a column, and they are mainly done by these engineers or these commercial suppliers who provide us the different types of columns that can be used for purification. Okay, so just understand this fact that it is L by H, and there are two other formulas. So if you want to express it in terms of, because you do not know the L and or just by looking at the chromatogram, if you want to have an idea of the number of theoretical plates, then you can use this formula 16 whole TR by W. So TR is the retention time divided by the peak width, or you can express it in terms of half peak width. Okay, so see this figure also. So this figure is taken from the book Wilson and Walker. So so here you see this example or see this figure. So there is one peak A and this is the second peak. Okay, so so the retention time of peak A is what? It is TRA. And the retention time of peak B is what? It is TRB. Okay, so these two analytes are having different peak retention times, that is TRA and TRB. Similarly, they are having different peak widths as well. So the peak width of A is WHA, peak width of B is WHB. Okay, and this is the half peak height, the height half height of the uh, half height is the half each row. Okay, so these different parameters you can use, and then you can get this number of theoretical plate value. So so that is what in this formula you were seeing, right? This TR by W. So W is basically the average of this WA and WB, and this one, this TR is the difference in this resolution or retention time of these two. Okay, so you can express it in terms of the retention time. You can express it in terms of the peak width as well. So if you see these two figures, so what do these two figures? So this is a well-resolved peak, isn't it? So this is well-resolved peak. So if there are if there are two figures, A and B, and you are asked that which of the column, which column has a better resolution, then obviously this has a better resolution because the two peaks are separate. While here you see that the two peaks have merged. Okay, the column has not been able to separate the two peaks as separate entities. Okay, there can be the other also. Sometimes you get a, a tailing effect. So tailing effect also means that there can be another population. So if these two peaks come very close to each other, then there can be a tailing which can be seen in the curve. Okay. So, you can read all this theory. So this so I'm not going to very detail how you got this different expression. Okay. So that will involve lot of calculations and stuff, but just understand the concept. And so the chromatographic columns are considered to consist of a number of adjacent zones in each of which there is sufficient space for an analyte to completely equilibrate between the two phases. So each zone is called a theoretical plate. So that is what I was explaining you through the figure. So it can be considered as having multiple theoretical plates. And n is the number of theoretical plates. So the higher the number of theoretical plates, the better is the resolution. Okay. So basically, so if from this expression, what you can say, so the higher the value of L, so the higher the length of the column. If you use a column which is having higher length, that will give you a better resolution compared to a column which is having a smaller length. Then another factor is H. H is what? The H is the height of this theoretical plate. Okay. So that is the number of theoretical plates in the whole column of length L is equal to L divided by the plate height. So just remember this or understand this concept. That is enough. Okay. So now resolution is what? So as I was explaining, L equal to L by H. So just so that is something for which you need to know the length, and also you need to know the height. But just looking at the chromatogram profile, you can see which is having a better resolution. So the one having a better the peak width. So the peak width and the peak height. So as the number of distribution plane will go up, it will allow the analyte to travel for longer period of time. Consequently, it will increase the distance between two peaks. So basically here, these theoretical plates have been called as distribution plate. So what will happen if the theoretical plates in number of theoretical plate increase? So if the number of theoretical plates increase, then it will allow the analyte to travel for longer period of time. And it will increase the distance between two peaks. The distance, so one you will get here and another you will get here. But if the number of theoretical plates are less, then you will the two peaks will be merged. Okay. So as the number of distribution plane will go up, it will reduce the width of the base of the peak. So if you look at these two peaks, so this is having a larger peak width compared to this one. Okay, that is why what is basically taken, the half of the peak width is taken for calculating the resolution. So as the number is increasing, the peak width is decreasing. So if you see these two peak widths, these two peaks, one this and another is this one, first and second, which is having a higher peak width. This second one is having a higher peak width. Okay, so as the number will increase, the peak width will decrease. Okay, this will have a lesser peak width. Hence, the number of distribution is an indirect way. So you can see this figure as well. So the peak width, the lower the peak width, the higher will be your N and better will be your resolution. Okay. So what is distribution coefficient? Now answer this question. So distribution coefficient, just now we learned that it is the distribution of a compound between two phases. So KD is basically distribution of compound in, or the concentration of the analyte in phase A divided by concentration in phase B. So that will determine whether or what will be the retention time. So your KD is also controlling the retention time of the compound. So if it is having a very high KD value, if it is having a very high affinity from your for your stationary phase, then it will be eluted later. And the one which is having a lesser affinity for the stationary phase, that will be eluted first. Okay. So now answer this question. The role of stationary phase in column chromatography is what? So it moves through the column with this mobile phase. No, the stationary phase is basically the matrix of the column. It is the resin. Okay, so that is fixed. That will not move with the column. So that depends on the composition of the column. Whether it is there are different names of this column, Cepha grill, Cepha dex, or uh, then there are high load. So there are different materials which forms the matrix of the column. Okay. Cephadex, which is dextrose, and another component which is conjugated together to form the matrix. Okay, so it serves as the buffer for the analyte. That is wrong. Buffer for the analyte will be done by the mobile phase. Your mobile phase will be determining what is the buffer of the analyte. Okay, it remains stationary and interacts with the analyte. So this is the correct answer, isn't it? So the your stationary phase is stationary. It is not moving with the It is not moving when your mobile phase is passing. It is stationary. It is the matrix of the column. Okay. And it will only interact with the analyte. Okay. And it will not evaporate during. So these all are wrong statements. Okay. So answer is option C. Now, what is resolution in chromatography? So just now we learned that resolution is basically the ability to separate two objects. Isn't it? Resolution we learned in microscopy as well. So resolution is basically the ability to separate two different points as two separate objects. Okay. Or in case of the chromatography, resolution is basically the ability to separate one peak from the other. To separate two analyte peaks. So the ability of a chromatographic column to separate two analyte peaks is resolution. What is retention volume? That is, so retention volume is, it can be retention time, it can be retention volume. That is at what time or at what volume of your mobile phase your analyte is getting eluted. That is your retention volume. So the volume or time it takes for an analyte to elute from the column. So what volume of buffer or what? So you can express it in both volume or it can be expressed in time, that what volume of buffer is required to elute your protein. Okay, that is your retention volume. So, okay, so for example, if you have a mixture of different proteins, so some protein will get eluted within 45 minutes, some will get eluted after 90 minutes. So that is the retention volume. So what are the factors that will affect the resolution in chromatography? The temperature of the mobile phase, the volume of the buffer use, the number of distribution events in the column. So just now we learned that this depends on the number of theoretical plates or number of distribution events in the column. So answer is basically number of distribution events in the column. So these are wrong false statements. Okay. Option C. The overlapping peaks in a chromatogram indicates what? If there are overlapping peaks, then it means that there is poor resolution. Isn't it? Poor separation of analytes. If there is overlapping peaks, or if the statement can come like if there is a tailing in the chromatogram profile, then also it indicates poor separation. Okay, so you must get a very sharp peak. So if it is a very if your column has a high resolution, then it will give a very sharp peak. Okay, if it's a sharp peak, and it must have a very less peak width. Peak width must be less. So now the protein coming to the chromatography. Okay. So till now we understood the basics of chromatography. So what is the principle of chromatography? What is resolution? What is retention time? Now you want to use this principle of chromatography to purify your protein. So your protein has different characters, isn't it? It has So your protein has different characters. It has an affinity, it has charge, it has surface area, it has hydrophobicity. Okay. So depending on why do different proteins have different charge? Because each protein are composed has different amino acid residues. That composes the protein molecule. So if your protein is composed of more number of acidic residues, then your protein has a negative charge. Isn't it? If your protein is composed of highly basic residues, then it will have a positive charge. Okay. If it has a large number of non-polar residues, then it will be hydrophobic. Okay. Now, if your protein is for example, if it is an antibody, then it will have affinity for antigen. Or if it is okay, if it has some fusion tag, then also it can have different affinity, and also it will have a surface area. Okay. Depending on what are the residues, which are amino acid residues, which are composed, or which constitutes the protein determined, that will determine the character of a protein. Okay. So depending on utilizing these different characters of a protein, like what is the charge, what is the hydrophobicity, what is the affinity, what is the surface area, we can use different chromatographic technique. So when we are utilizing the charge of a protein, just we are considering on the basis of charge. If we are trying to separate protein, then that will be called as ion exchange chromatography. If we are if we are purifying protein, or if we are separating our protein molecules on the basis of hydrophobicity, then that is hydrophobic interaction chromatography. If it is on the basis of affinity, then we can use affinity chromatography. Or if it is on the basis of size or surface area, then that is called the gel filtration chromatography, or it is also called as size exclusion chromatography. Okay. So the protein has a very dynamic personality. All proteins have different, uh, each protein are unique. Each protein is unique, and each protein will have a particular charge, it will have a particular shape, it will have a particular size, surface area. So utilizing this differences in the proteins, we can isolate our protein or purify our protein of interest. So ion exchange chromatography is what? So ion exchange chromatography, when we are separating our protein of interest on the basis of charge. Okay, so the chromatography distributes the analyte molecule as per charge and their affinity towards the oppositely charged matrix. The analytes bound to the matrix are exchanged with a competitive counter ion to elute. The interaction between matrix and analyte is determined by net charge, ionic strength, and pH of the buffer. So what is basically done in ion exchange chromatography is basically there are different matrices. Okay, there are different matrices that are commercial. These are commercially available. Okay, so you can either have a cation exchange chromatography column. Okay, so you can have a cation exchange chromatography column, or you can have an anion exchange chromatography column. So what is the meaning of a cation exchange column and an anion exchange anion exchange column? So basically your anion exchange column will have a anion exchange column has a positive charge. Okay, the matrix will be positively charged, so they will attract molecules which are having negative charges. Okay, and cation exchange column will have negative charge. The matrix will be negative charge, so it will attract molecules which are positively charged. So depending on what is your protein of interest, accordingly you can use a cation exchange column or you can use an anion exchange column. Okay, so for example, you have two protein. Okay, yeah, you are for example you want to purify a protein which is highly basic. Okay, which is highly basic. So if it is highly basic, with what does it mean? That its PI is very high. PI is very high. PI is very high. Okay. So for example, proteins which are highly basic, they have a PI of around 10.8. Okay. So at such a high PI, what will happen? The normal physiological buffers that we use, like for example, we are using tris buffer or HIPIS buffer, or say we are using uh, any sodium acetate buffer. Then these different buffers at these pH, what will happen? Your protein will be positively charged. So what is the basic principle? That if the pH is less than the PI of the protein, so if the pH of the buffer is less than the PI of the protein, then your protein will be positively charged. We all know this basic biochemistry, isn't it? But if the pH of the buffer is greater than the PI of the protein, then your protein will be negatively charged. Negatively charged. Okay. So depending on what is your the protein, what is your protein of interest? If you are trying to purify a very basic protein, say for example, you are trying to purify histone proteins, then your histone proteins, they are very rich in lysine arginine residues. So they have positive charge. So as they are rich in positive charge, so you will basically use a cation exchange chromatograph. We'll use a cation exchange chromatography column. Okay. Or for example, you are trying to purify very certain acidic protein. Okay, whose pI is very low, very low. Then what you can use? Then you can use an anion exchange column, because the physiological pH buffers that we normally use in laboratory, PBS buffer or your trace buffer, they will have pH 7 around 7.5. So at this pH, your protein will be, in case of acidic protein, it will be negatively charged. So you can use the anion exchange chromatography column. Okay. But if your protein is is highly basic, then it will be positively charged and you can use cation exchange chromatography column. Okay. So you see, so they see this figure carefully. So this orange color structure, this is your column. This is your column. This is the matrix of the column, and your matrix of the column can either be positively charged. So here in this case, it is positively charged. So if the matrix is positively charged, then what will it do? It will attract negatively charged species. Okay. Now your analyte has different molecules. It can have doubly charged negative ions. It can have singly charged negative ions. It can be neutral. It can be positively charged. So if your analyte has all these different molecules, one is having M plus, another is having M0, another is having M2 minus, another is having M minus. Then if you pass all these, then which of these molecules will have higher affinity? The one which is having -2 charge, that will have more affinity for the matrix compared to the one which is having positive charge. So the one which is having positive charge, that will get eluted very fast. Okay, that will be eluted. That will not interact with the column. It will be eluted. Okay. When you pass the mobile phase, it will come out with the mobile phase. Okay. But the one which is having the negative charge, 2 minus, or the neutral charge, one which is having neutral charge, that will also not interact. So at very lower elution volume or the retention volume that I was discussing, that low retention volume, your M plus and M0 will get eluted. Now, as you now for eluting this negatively charged species like 2 minus and M minus, you need to increase the salt concentration. Okay, the salt concentration needs to be increased so that these higher negative charge species are eluted. Okay, so this is basically a cation exchange column. No, this is an anion exchange column. Okay, so if you are given this figure and you are asked that is this an example of cation exchange column or anion exchange column? So remember, your anion exchange column means your column is attracting negatively charged species. So if your column is attracting negatively charged species, it means that your column is positively charged. Okay. And if it is a cation exchange column, cation exchange means it will attract positively charged species. Okay, positively charged species. It means that the column, the matrix of the column will be negatively charged. Okay, do not get confused. Cation exchange means your analyte will be positively charged, but your matrix has to be negatively charged, only then it can attract the positively charged species. Okay. Now, there are different types, as I was explaining. So, this cation exchange chromatography, the resin which characterizes or which constitutes this cation exchange chromatography column, they will have what? They will have negative charge. So negative charge means it must have some negative charge functional groups. Like you see here, this OCH to COOH, this is negative charge, isn't it? So if this H is ionizable and it will be COO minus. So carboxymethyl. So anything like carboxy, if it is a carboxy group, or if it is a sulfopropyl, your sulfate group is also negatively charged, isn't it? SO4 2 minus. Or it is a sulfonate. Okay, then there can be these molecules, like carboxy, sulfopropyl, sulfonate, all these are what? All these are negatively charged species. So they are all cation exchange columns. These are cation exchange columns. But if it is an anion exchange column, then your column will be what? Your column will be positively charged. Positively charged means it must have some functional groups like amine or quaternary amine, all these things when they are present, like diethyl amino. So whenever you see this amino group, or quaternary amino. So all these groups, if they are present, then it will be positively charged. Your column will be positively charged, but it will attract negatively charged species. That's why it is called anion exchanger. Okay. So do not get confused. So try to remember this. Sometimes this type of question comes that you are given a name and you are told to identify that whether it is a cation exchange or an anion exchange column. So you do not have to memorize. If you understand the concept, you will get the question right. Okay. So if it is anion exchange means first, so if it is an anion exchange, then you have to remember that your column will be positively charged. And which functional groups give positive charge? It is amine groups. Okay. So anything like amino, quaternary amine, all these terms, if they are written, then it will be anion exchanger. And if anything like carboxy or sulfonyl or sulfonate, all these are written, it means that the matrix is negatively charged, which means that it is a cation exchanger. Okay. So, yeah, you can read this text. Okay. So the whatever I was explaining, that is written as well, and it was explained in the lecture as well. Okay. So you see this figure carefully. So if it is a cation exchange chromatography, then your matrix is what? Your matrix is negatively charged. Okay. Matrix is negatively charged. So it will attract the positively charged species. These proteins which are positively charged, that will be attracted. Okay. So now if you want to elute this protein, then what you have to do? You have to increase the salt concentration. So once you increase the salt concentration, then at very high, at a particular concentration, what will happen? If you are increasing the salt concentration, means you increasing the NaCl concentration. So at high NaCl concentration, your sodium ions will uh, compete with the protein. with the protein. Your protein is positively charged and your sodium ion is also positively charged. So your protein and your sodium ions, they will compete with each other, and at a particular concentration, this sodium ion will not allow the protein the positively charged protein to bind to its bind to the matrix. Rather, it itself will go and bind to the matrix, and your protein will get eluted. Okay. Okay, so you see this figure. So first there is binding. So in any ion exchange chromatography, there are three steps basically. One is equilibration. Okay, there are three main steps. Like equilibration, then there is sample injection. So if you have used a any chromato chromatography instrument, then you must, and if you have done an ion exchange chromatography, then you will know that there are four basic steps. One is equilibration, then there is sample injection, then there is column wash. Okay, column wash is done to remove the non-specific binding. Then there is elution, or we can also call this fractionation. And in the last step, there is regeneration. Okay, so in the first step, in the sample injection step, there will be binding. Basically, your analyte will go and bind to your column. And in the column wash step, you are not increasing the salt concentration. You are just keeping the same salt concentration, and you are removing the unbound species. Like if there is a neutral molecule, so that will not interact with the column. It will get eluted. So all these non-specific binding will be eluted, and then in the elution step, you increase the salt concentration. So like here you see this figure. Once you are increasing the salt concentration, your salt will compete with your protein molecule, and it will remove, or it will not allow the analyte molecule to remain bound to the matrix. Rather, this molecule itself will go and bind to your matrix. Okay, and this way, in this elution step, your protein will get eluted. While in anion exchange chromatography, your matrix is positively charged. Okay. Your matrix is positively charged. Okay. So all the negatively charged protein molecules will bind to it. But when you are increasing the salt molecule, the salt concentration, your chloride will compete with this protein molecule, and it will remove the bound protein molecule from the matrix, and it will itself will go and bind to the matrix. Okay. This is all. So now what are the different factors? So as I was explaining initially, so in ion exchange chromatography, you need to know that what is the PI of your protein, whether your protein is an acidic protein, whether it is a basic protein. Accordingly, you have to choose the buffer. Okay. So if it is an acidic protein, then what column will you use? You need to understand this concept. If it is an acidic protein, it means that the protein at normal, at physiological pH it will be what? It will be negatively charged. So you need to use an anion exchange chromatography column. So you need to use anion exchange. Okay. While if your PI is very high, PI is high means if your PI is high means it is a basic protein. Okay. So for basic protein, what type of column do you use? It will be your basic protein means your protein will be positively charged at pH 7.4. So you will use a cation exchange column. So these type of questions can come, and you have to understand the concept. Otherwise, you will get confused. So that is what is explained here. So if your PI is positive, which is the case for basic proteins, then it will be a cation exchange chromatography. If your protein is negatively charged, then you can go for anion exchange chromatography. Okay. PI and the net charge below the PI, anion exchange you use above the PI value. So what will happen if you use a buffer having a pH which is equal to the PI of the protein? Anyone? So if you use a buffer which is having a pH equal to the PI of the protein, then in that case your protein will have zero charge, isn't it? Your protein will have zero charge at a pH equal to the PI of the protein, and in that case your protein will aggregate. It will not be soluble in the buffer. Okay, so you either need to choose a pH which is below the pI, or you need to choose a pH which is above the pI of the protein. Only then you can elute your protein. Only then your protein will be soluble. You also need to know the pH of the protein. So pH of the buffer is also you have to pH is also important, isn't it? Because the pH will determine what will be the charge on the protein. So if your pH is less than pI, it will be positively charged. If your pH is less than pI, and if your pH is greater than pI, then what will happen? So at pH less than pI, your protein will be positively charged. Will be positively charged, and if your pH is greater than pI, then your protein will be negatively charged. Okay. So there is one tool. So you can directly write on the web Expressi prot param. Expressi, you can use prot param. You can directly type on the Google, and then you will see that a file, this server will open, and then you can just put the protein sequence. So there will be a text. So you see this text box. So in this text box, you put the protein sequence, and then you click on compute. Click here to compute PI or PI or molecular weight. You just click here, and this software, this server will give you all the different parameters of your proteins. What is the PI of your protein? What is the molecular weight of your protein? So if you go to plot param, you can have all the different idea of all the parameters which are present in your protein. So what is the PI of your protein? What is the molecular weight of your protein? How many aromatic amino acids are there in your protein? How many basic amino acids are there? How many acidic amino acid residues are there? Then what is the hydrophobicity of your protein? All these, what are the number of amino acid residues? All these different information you will get in prot param, or otherwise you can use this server also if you just want to and get the PI and molecular weight. You just expressly compute PI or molecular. So understand this: at pH equal to PI, net charge will be 0. Okay. You can use, so this link has been provided. You can go and check what is the PI of your protein. Otherwise, you can calculate also. So depending on the amino acid residues that are there, you can also calculate the PI of your protein. Okay, what is the pH? Each of your buffer, each of the amino acid residues will either be positively charged or negatively charged. And accordingly, you can get the net charge of the protein. And also your PI is also, you can get from the pKa values, pKa1 plus pKa2 by 2. Using this value also, you can get the PI value. Okay. So if it is a very large protein, if they're having several amino acid residues, then it is better to use a software. Okay, so how does this ion exchange chromatography work? So you see here, so you will, if you, so the chromatogram profile, how will it look? So you will have a first peak, you are getting 3 peaks. So the first peak will correspond to your positively charged. So if you are using an anion exchange column, so here you have used an anion exchange column. As a result, what has happened? Because it is an anion exchange column, your matrix is positively charged. So your matrix will not interact with the positively charged and this neutral species. As a result, your positively charged and neutral species are getting eluted earlier compared to the higher negatively charged species. Okay. So when you're doing a chromatography or you're setting up setting up all the buffer for performing the chromatography, you must ensure that your buffers have been filtered. There are no impurities. It has been sonicated, and so you have to ensure all these things so that there is no clogging of the column. If your column gets clogged, then it will generate a very high back pressure. Your column pressure will increase, and your column can break. Okay. So you have to check that that all these you have filtered the different analyte. You have also filtered the mobile phase, and then you are setting it up. Okay. So the mobile phase, when you are preparing the mobile phase, what your pH, you should measure the pH of the mobile phase. So for ion exchange chromatography, it is very essential that you measure the pH of the mobile phase, because depending on what is the pH of your mobile phase, that will control that what is the pH of your protein. So the more the difference between the pH and pI of the protein, the more will be the affinity. Okay. So sample preparation, it should be prepared. You can prepare it in mobile phase. Usually it is prepared in the mobile phase, and it should also be filtered so that it is free of any. So if your protein has aggregated, then what we usually do, we spin the protein at 13,000 RPM. We remove the precipitate, and then we take the supernatant and inject it in the column. Okay, and then elution. So there are two ways of elution. So for ion exchange chromatography, what is done is that a gradient elution is done. So a particular salt concentration is used, and then after a particular volume, you increase the salt concentration gradually. Okay, so from 30% NaCl to, say, for example, like 80% NaCl, and you keep the column volume about 100 ml. So in this 100 ml, the percentage. So basically, in ion exchange chromatography, you have two mobile phases. One you keep a mobile phase A, and another you keep a mobile phase B. So your mobile phase has low salt concentration, and your mobile phase B has high salt concentration. Okay, high salt concentration. Then in the elution step, what is done is that the concentration of B is gradually increased. Okay, so you have pipes coming out from this both A tubes coming out from both A and B, and then there is a pump, and then there is a mixer. Okay, so this mixer will allow a particular concentration of A to mix with a particular concentration of B, and accordingly you will get a conductivity. So depending on the conductivity, your protein will be. So high conductivity means high salt concentration, isn't it? So as you increase the salt concentration, the one which is having a one which is very tightly bound to your protein or your protein of interest will get eluted later. Okay. Accordingly, you have to set the mobile phase and all these A and B buffer. What is the salt concentration? What is the pH? Everything you have to set. Okay. Now column regeneration, what is done? So in column regeneration, we apply a very high concentration of salt. So at very high concentration, so 100% B, we set it to 100% B. So at 100% B, for example, your B is having a salt concentration of 2 molar NaCl, and at such a high concentration, all the species which are bound to your column and which have not eluted, or all your non-specific binding, non-specific bound protein, they will all get eluted. Okay. So what are the different applications of ion exchange chromatography? Then the application is like here in this case. So if you want to purify DNA, so your DNA is basically negatively charged. Okay. So for negatively charged species, what do we use? For negatively charged species, we use anion exchange chromatography. For positively charged species, we use cation exchange chromatography. Only this much is required to understand. Okay. So negatively charged, then we are using anion exchange chromatography. So for example, you want to separate the DNA, and your DNA, because it is negatively charged, it will remain bound to some protein. You want to isolate the DNA from the protein. Then what you can use, you can inject it in an anion exchange chromatography column. Your DNA will remain bound to your column while all your proteins will get diluted. Okay. Then at high concentration of salt, your DNA will also get diluted. And you can collect the fraction of DNA, you can concentrate it, and then you can use it in further downstream processes. Okay. You see here your protein X is getting eluted in the flow through. And while your the DNA molecule, because your matrix is positively charged, your DNA molecule will remain bound to your beads. So you see here it is bound to your beads while your protein X is getting eluted. Okay. So you can purify your DNA using anion exchange chromatography column. Okay. Now, what is the application of the another application is using this protein kinase assay. So, what is this protein kinase assay? So, you can set up a small biochemical reaction. So, you have taken two tubes. Okay, you have taken two tubes. In one of the tubes, you have added some kinase protein. So, casein kinase you have added. Okay, you have added this casein kinase. You have added the substrate. Okay. And in another one, what you have done, you have added this kinase enzyme, CK. CK enzyme you have added, but you have not added the substrate. Or say, for example, you have just added the substrate you have added, but the enzyme you have not added. Okay. And now, so if you add the substrate and you add the enzyme, then what will happen? Your substrate will get phosphorylated. While in this case, your substrate will not be phosphorylated. So, for example, you want to separate a phosphorylated protein from a non-phosphorylated protein. Then also your phosphorylated will be negatively charged, isn't it? Your for 3 minus, they will have negative charge. So as a result, your this negatively charged species, they will. So if you are using an anion exchange chromatography column, then your phosphorylated substrate will remain bound to your species, while your non-phosphorylated will get eluted. Okay. So this is basically what they have done. They have radio labeled this substrate with C14. C14, and then they have set up this kinase reaction. So they have set up the kinase reaction, so their substrate will get phosphorylated. Okay, so you see here this substrate is having this PO4 3 minus group added to it. Okay, so in the flow 3, you can get the radioactive count. While in the control reaction, you have not added this enzyme, so your substrate is not getting phosphorylated. And as a result, your here basically you have used a cation exchange chromatography column. To separate, they have used a cation exchange chromatography column. So what will happen if you use a cation exchange chromatography column? So if you use a cation exchange chromatography column, then your protein, because the then what will happen? The phosphorylated protein will be eluted in the flow through. Okay, while the non-phosphorylated protein will remain bound. Non-phosphorylated will remain bound. So depending on what column you are using, that will determine which will get eluted. So if it is a cation exchange chromatography column, then your non-phosphorylated will this will remain bound. Negatively charged will remain bound. The phosphorylated will remain bound, and your non-phosphorylated will get eluted. This will get eluted. So because it is a cation exchange column, so cation exchange column, what will it do? It will bind only positively charged species, isn't it? Your cation exchange column will bind positively charged species. Okay. So your phosphorylated one will be eluted. Okay. Sorry for the confusion. Actually, you are using a cation exchange column, isn't it? So basically, if you use a cation exchange column, so the cation exchange column will be, it means the column is negatively charged. It will bind to positively charged species. If it binds to positively charged species, then what will happen? Your phosphorylated protein will get eluted. Phosphorylated protein will get eluted. Non-phosphorylated will remain bound. Okay. So in the flow through, you can detect, you can quantify this, that what is the what is the radioactive count that will be equal to the phosphorylation rate of the enzyme that you can do. Okay, assay you can do protein kinase assay, that what is the rate of phosphorylation that you can get by measuring the this radioactivity count in the flow through. Okay. Now softening of hard of water. So this hard waters have different ions, isn't it? They have the doubly charged positively doubly charged positive ions like calcium, magnesium, and other cationic metals. So if you have a Ca2 plus ion, even if you have a Na plus ion, and if you use a cation exchange column, then what will happen? The cation exchange column will bind more efficiently to your cation exchange column will bind more efficiently to your calcium ion than your sodium ion. So so you have your water. You have the water. The water has all these different ions. It has calcium, sodium, magnesium. Then you when you inject this water in this cation exchange column, then what will happen? All these positively charged and calcium, magnesium, which makes the water hard. The water hard water means what? They're having very high concentration of this calcium, magnesium salts. So all these salts will get bound to your column. Okay, while this positively charged species, they will get eluted. Okay, so as a result, using this, you can convert a hard water into salt water because all the this calcium, magnesium salt, they will remain bound to your column, while this positively charged, they will get eluted, and now you can yeah, further remove the sodium by some other purification method. Okay. So now hydrophobic interaction chromatography is basically utilizing the hydrophobicity of the protein for purification. So hydrophobic interaction chromatography exploits the ability of a strong interaction between a hydrophobic group attached to the matrix and the hydrophobic patches present on an analyte such as protein. So this is the hydrophobic patches. So the dark red colored this loop or this structure is the part. This is the part of the protein which is highly hydrophobic. Means it has several non-polar residues. Okay. So what will happen? These hydrophobic residues will have like the concept of like attracts like, isn't it? So if it is hydrophobic, then it will go and bind to the hydrophobic portion of your matrix, and the one which is less hydrophobic, that will be eluted first. Okay. So the hydrophobic group attached the matrix is made hydrophobic. So see this. So hydrophobic means your matrix has these different hydrocarbon chains, octyl group, or they have this hexane group, this six hydrocarbon chains, or they can have 9 or 12 hydrocarbon chains. So the more the hydrophobic the protein, the more strongly it will bind to the column, and the one which is less hydrophobic, it will get eluted first. Okay, so that is the basic concept. Now salting out and salting in yeah, is the principle by which this HIC works, hydrophobic interaction chromatography. So what is salting out and salting in? So basically, when you are increasing the salt concentration, so till a particular salt concentration, your protein is soluble. Now, the protein is soluble, but once you start going beyond that particular salt concentration, your protein starts precipitating. Okay, so because the protein contains multiple charge group, its solubility depends on the concentration of dissolved salts, the polarity of the solvent, the pH, and the temperature. The solubility of a protein at low ion concentration increases as the salt is added. So as you will increase the salt concentration, the solubility of protein increase. Solubility of the protein will increase, but it will only increase to a particular salt concentration. So that phenomenon is called the salting in. While so the additional ions shield the protein's multiple ionic charges, thereby weakening the attractive forces between individual protein molecules. So what happens is basically your proteins, if your protein molecules have positive charge, then these different protein molecules that are present, they will face repulsion with each other. Okay, therefore, so your protein has it is a basic protein, say for example, then it has multiple positive charges. So these protein molecules will tend to repel each other. So what happens that when you add salt, so for example, you are increasing the salt concentration. So till a particular salt concentration, this repulsion that is there between this protein molecule that will be reduced. So your protein becomes soluble till a particular salt concentration. But when you go beyond that salt concentration, you are keeping on increasing the salt concentration. Then after that particular salt concentration, your protein will, what will happen? That the hydrophobic residues that are there, they will now tend to be exposed. Okay. However, as more salt is added, particularly with sulfate salts, the solubility of the protein again decreases. So it basically has this type of graph. So till a particular concentration, your your solubility increases, but after that concentration, your solubility decreases. The sorting out effect is primarily as a result of competition between the added salt ions and the other dissolved solutes for solvent molecules. So at very high salt concentration, many of the added ions are solvated and there is significantly. So this now this what happens once you are increasing the salt concentration. Now this Cl minus, they will start forming a water molecule will start forming a cage around these ions. So this H, these so they will. So these ions get solvated, okay, and the protein molecules, the water molecules no longer solvate this protein molecules. Rather, the water molecules will solvate the ions, and their solubility will decrease. Okay, so there is less bulk solvent available to dissolve substances, including proteins. So anything is soluble. Why? Because what happens? This water molecules or shell of water is formed around this charged molecules. So this negative positive, and it makes the molecule soluble. Okay, now this is ammonium sulfate. Uh, Now, so what happens? So this is a figure taken from the book Voigt and Voigt Biochemistry. So you can see here that the salt of choice, usually ammonium sulfate, is added to a solution of macromolecules. So you have a solution of macromolecules where you have different protein molecules. This red colored, yellow colored, green colored, they are different protein molecules. Now you are keeping on adding ammonium sulfate. Now what happens at a particular ammonium sulfate concentration? Your red protein molecules, they first precipitate. Okay. Now, once they are precipitated, you can spin and you can obtain the supernatant. Now, again, what you do, you are now again increasing the ammonium sulfate concentration. Now, at this particular ammonium sulfate concentration, your green protein molecules, they get precipitated, and you are left with your with the pure yellow protein molecules. Okay, so that is the principle that is followed by ammonium sulfate method for purification of protein. So yeah, each protein has a particular concentration, or there has a it has a particular ammonium. So at a particular ammonium sulfate concentration, each protein gets precipitated. So there can be a protein that gets precipitated at 0.5 molar ammonium sulfate. There can be a protein which gets eluted at 3 molar ammonium salt concentration. So accordingly, you can vary this and you can obtain the pure protein. So what is the choice of HIC matrix? So if you see this HIC matrices, they are all these having this butyl, octyl, phenyl. So these different the hydrophobic groups. So all these hydrophobic groups or hydrocarbon chains constitute the hydrophobic interaction chromatography matrix. While in case of ion exchange, they were all charged, like carboxy or sulfur or amine. So they are all charged matrices. But here you have hydrophobic matrices. So the functional group is butyl, phenyl octyl. So these are the different functional groups. Okay. Now, what is the principle of HIC? So HIC for basically it follows the principle that hydrophobic interactions between protein and the chromatographic matrix can be exploited to purify the protein. So basically, it is following the principle of like attracts like. So the more hydrophobic your matrix is hydrophobic, your protein is hydrophobic. So more hydrophobic the protein, the more strongly it will be attracted to your matrix. So in hydrophobic interaction chromatography, the matrix material is likely substituted with octyl or phenyl groups. So at high salt concentration, what happens? So what will happen at high salt concentration? When the salt concentration is very high. So at high salt concentration, your hydrophobic proteins will be more strongly bound. Okay, hydrophobic proteins will be more strongly bound. Because at high salt concentration, the salting out principle is taking place. Your hydrophobic protein will, okay, hydrophobic proteins will be strongly bound. Now, so the binding will take place in high salt concentration. Binding of protein will take place in high salt concentration. At high salt concentration. Now, for because at high salt concentration, only the hydrophobic proteins will remain bound to your matrix. Now, as you decrease the salt concentration, so for elution, what you do? So this is actually the reverse of ion exchange chromatography. In ion exchange chromatography, you were increasing the salt concentration for elution of your protein. In hydrophobic interaction chromatography, you are decreasing the salt concentration. Okay, you will decrease the salt concentration. So once you decrease the salt concentration, what will happen? First, the most hydrophilic protein will be eluted and later the most hydrophobic will be eluted. Okay, so the eluent is typically an aqueous buffer with decreasing salt concentration. So remember this. I hope this concept is clear. So in hydrophobic interaction, the binding is taking place in high salt concentration and then you're gradually decreasing the salt concentration. So at low salt concentration, the most hydrophobic protein will get eluted. And at high salt concentration, the least hydrophobic protein will get eluted. Okay. So you see this figure also. So this is high salt. So at high salt, your protein is remaining bound to the matrix. Now, once you are lowering the salt concentration, your protein will get eluted and your matrix will become free. So the equilibration is done at high salt concentration at 0.5 to 1.5 molar sodium sulfate. So this salt must be below the concentration where it has salting out effect. Okay. Salting the concentration at which salting out is taking place. So at that concentration, your protein will get precipitated. So you have to keep the concentration of the salt below the concentration at which salting out is taking place. Okay, because at this concentration, your hydrophobic patches of the protein will get exposed and they will remain bound to your matrix. Okay. So sample is prepared in mobile phase and it should be free of suspended particle to avoid clogging of the column. The most recommended method to apply the sample is to inject the sample with a syringe. Okay. You have to the sample that you are injecting, either you have to filter it or you have to spin it to remove the precipitate that is present. Okay. And elution, you will elute it at low salt concentration. So by decreasing the salt concentration, you can elute, or you can either change the polarity of the mobile phase, such as alcohol, or you can use a detergent to displace the bound protein. So either you decrease the salt concentration is one method, or you increase the polarity. What will you do? Polarity. So if you on the basis of polarity you have to do, then what will happen? So at low polarity, so if you increase the polarity, then what will happen? Answer. If you increase the polarity of the buffer, then what will happen? So if you increase the polarity of the buffer, then your hydrophobic at low polarity, your protein will get eluted. The most hydrophobic protein will get eluted at low polarity, at less polarity, and the least hydrophobic protein will get eluted at higher polarity. Okay. So the other is you can use also detergent. Now the next step, last step is column regeneration. When you have got your protein, you pass either if you are on the basis of polarity you are doing or on the basis of salt you are doing. You have to apply a very low salt, or you have to apply a very high, very non-polar solvent. So yeah, that your protein, all the non-bound proteins get diluted. Okay, so sorry for this. I'll just sorry. The theoretical PI of the protein is calculated by what? So it will depend on the individual amino acids and their pKa values. Okay, so what is the pKa value? That will depend that. So PI is basically pKa1 plus pKa2 by 2. That is option C. The online tool that can be used for experimental calculation of a protein's PI is what? It is Expasis Compute PI. Expasis Prot Param or Expasis Control Compute PI is the answer. Now the illusion, okay. The illusion typically achieved in ion exchange chromatography by what? So in ion exchange chromatography, we basically increase the salt concentration. The salt concentration has to be increased for ion exchange chromatography using stepwise or continuous gradient of salt. Stepwise means what? First, you are doing from 20% to 30%, and then after 30%, you are doing from 30 to 35%. Then from 35 to percent, you are going to 65%. So this is stepwise increase in salt concentration. And continuous means you have applied from 20% to 80%. This is continuous increase in gradient of the salt. So either you can do stepwise increase in salt, increase in gradient of salt, or you can do continuous increase or continuous gradient of salt. Okay. So the column material that is used to equilibrate in hydrophobic interaction is what? So in hydrophobic interaction, the column material is hydrophobic basically. So the column material that is used to the buffer containing ammonium sulfate is the answer. Okay, 0.15 molar ammonium sulfate to 1.5 molar ammonium sulfate is used for equilibrating your column, and this salt concentration should be the salt concentration which is below the concentration at which sorting out takes place. Okay, so that is all for today. So I hope you could understand the concept that I discussed today. So if you have any more doubts, we can take up those doubts in the next class. So thank you all for your patient listening.